Nick Majerison here. Welcome to Top Med Talk. Thanks for downloading the podcast. If you like what you hear, make sure you subscribe. This week we're presenting to you our Xtreme Everest mini-series, which we streamed on the website live from Duke University in America on June the 14th and the 15th when these presentations first took place. Now, this presentation is called Cardiovascular Adaptation to Hypoxia, and it's Daniel Martin. Have a listen.
Top Med Talk.
It's a great privilege to be here. Thank you very much for the invitation to speak. My task over the next 20 minutes or so is to talk about cardiovascular adaptation to hypoxia at high altitude and to sort of punctuate that with some of the studies that we've been doing over the last 15 years or so now at high altitude looking at this particular area. So I do a lot of undergraduate physiology teaching at UCL, so I try to keep things fairly simple for the students of today. So I talk about the cardiovascular system as having a sort of wiring component to it, controlling the overall function of the system, a big pump in the center which provides pressure and as a consequence of that flow, and some pipes circulating around the contents, so your blood. You may recognise that it's quite similar to your domestic heating system that you would have at home. With the differences, though, or at least what I think to be the primary difference between the two systems, is that the human cardiovascular system has to adapt to enormous changes both within and outside of the body to continue to provide our organs with the oxygen that we require. One of the other differences between your heating system and your cardiovascular system is that we've got something hidden away in every organ of the body called the microcirculation. And I'm going to come back to the microcirculation at the end of the talk, but the microcirculation is crucial to the delivery of oxygen and nutrients to every cell in your body. And even if you have a perfectly functioning macrocirculation, it's basically a complete waste of time if your microcirculation is dysfunctional. Just to quickly summarize what you've already heard about adaptation and physiology to hypoxia at high altitude. These are data taken from a chamber study in the 1980s called Operation Everest 2, where they put 6 or so subjects into a hyperbaric chamber over the course of about 40 days to simulate a journey to the summit of Everest. And here you can see in the blue line the oxygen cascade that Denny mentioned, so this movement of oxygen from the air that you breathe at the mouth right the way down to the tissue level to the mitochondria. If you then take people up to an altitude of about 8,000 meters in the chamber, you can see this flattening of the oxygen cascade and the removal of this pressure differential which drive oxygen through the system. There are 3 primary ways from an organ perspective that we adapt to high altitude. So there's an increase in respiratory rate and tidal volume, an increase in heart rate and cardiac output, and the increase in hemoglobin. And just once again, to stress this paradox that confuses people sometimes, this increased arterial oxygen content that we find at altitude. So there's more oxygen physically, convectively going around the system than you have at sea level. But despite that, we still suffer from hypoxia. So from an organ system perspective, the place where hypoxemia is detected is in the peripheral chemoreceptors of the carotid bodies. They send out signals to the respiratory center in the medulla, which causes an increase in both respiratory rate and depth. And there's also a spillover from the respiratory centers into the sympathetic nervous system, causing an increase in sympathetic drive. You're all very familiar, I'm sure, with what your sympathetic nervous system does, but these are the key points which are important to adaptation to high altitude related to the sympathetic nervous system. And this is not a system that is down-regulated over time at high altitude. This continues for the entire time that you're exposed to hypoxia. So you can measure both in plasma and urine the increase in catecholamines, both adrenaline and noradrenaline. It causes increase in inotropy and chronotropy. There's an increase in your cardiac output, not only at rest but whilst exercising. Systemic vasoconstriction, which we'll come back to in a little bit. An increase in blood pressure, although this is also slightly controversial. And an increase in pulmonary artery pressure, although this is not the primary cause of that. It certainly contributes to it. These are some old data now. This is about 40 years old, data collected from Pikes Peak, about 4,500 meters, and you can see this early rise in cardiac output and heart rate when you're immediately exposed to high altitude, which settles down a bit as over the days that you're there. But I think what you'll notice is the striking thing of interest here is that your stroke volume progressively falls whilst you're at altitude. And that is thought to be due to a reduction in circulating volume caused by diuresis that's inevitable at altitude. These are some data of our own, just very simple data, just to illuminate a few points. This is a combination of data from different expeditions we've been on, so over 300 of our subjects ascending to Everest Base Camp at 5,300 meters over a period of 11 days or so. And you can see this reduction in oxygen saturation down to about 80% or so by the time that people get to Everest Base Camp. And this further reduction on exercise. Now, this is not the exercise that you've heard Denny talking about in the previous talk, where people are exercising to their maximum capacity. This is a simple exercise test, just stepping on and off of a step for a couple of minutes. And you see consistently, whilst you're at altitude, this reduction in oxygen saturation as you exercise due to pulmonary diffusion limitation. You can see this steady rise in blood pressure sustained throughout the time of the expedition. And I said previously that it was perhaps controversial. There are some studies that have reported no rise in blood pressure, others which have reported an early fall in blood pressure. So there's certainly variability in the literature that's out there. We have always seen a reasonably sustained rise in blood pressure, certainly in our subjects. So sustained in some cases, though, that it may cause harm. We reported on one of our participants who came to Everest Base Camp, actually one of our investigators, a previously fit and well 30-year-old male who developed severe hypertension at altitude. You can see here his systolic blood pressure is the red line on these graphs, consistently getting higher and higher, peaking at about 223 over 119 millimeters of mercury, and on descent was seen to have an acute kidney injury, presumably because of that high blood pressure. But this individual did not go on to have hypertension when at sea level, and all of his investigations afterwards, including cardiac MRI, were completely normal. So clearly just very sensitive to that increase in sympathetic drive at altitude. Heart rate also rises because of this sympathetic stimulation. And here we just see something which I don't really have time to go into today, but hints towards something else that's been repeatedly reported at high altitude, which is this reduction in maximum heart rate that is seen when you're exercising. And again, I stress this is not maximal exercise that these people are enduring in this test, but definitely in other experiments we see this fall in maximum heart rate as a result of ascent. I'm just going to go on to the circulatory system now, and given exposure to acute hypoxia, I've explained the sympathetic activation that occurs, but there's also some degree of systemic vasodilatation. So we have what you call vascular antagonism. So there's both vasodilatation and vasoconstriction, perhaps with the vasodilatation occurring extremely quickly and then profound and constant vasoconstriction after that. And the molecule that is almost certainly responsible for that is nitric oxide. So formed in the endothelium, it causes profound vasodilatation. And we have also looked at nitric oxide metabolism at high altitude. And here you can see nitrate, nitrite, and S-nitrosothiols levels increasing in a cohort of participants trekking to Everest Base Camp. And along with that, this increase in cyclic GMP activity, showing you a sort of upregulation of the whole nitric oxide system. Not only does that have an effect on your vascular system, but as you may well hear in the next talk, that it has some very subtle metabolic effects also. The pulmonary circulation works in quite a different way to the systemic circulation, as I'm sure you understand. And what we see at altitude is a sustained hypoxic pulmonary vasoconstriction. And this is because of an increase in calcium release in the vessels in the pulmonary circulation, along with a reduction in nitric oxide because of the lack of oxygen there, and this increase in sympathetic nervous system that I mentioned before, which causes a raised pulmonary artery pressure. Again, these are data from that experiment in the hypobaric chamber, the Operation Everest 2, and what you can see on the left is subjects exercising, so they're at rest on the far left of the graph where their cardiac output is approximately sort of 5 litres a minute or so, and as they exercise you can see at sea level their pulmonary artery pressure rises. The middle graph is now up to about 6,000 meters in the chamber, and already you can see that the pulmonary artery pressure at rest is about as high as it gets at exercise. during sea level, and further at 7,620 meters, really quite extreme increases in pulmonary artery pressure at these altitudes. Perhaps one of the interesting things about this raised pulmonary artery pressure is what happens afterwards. And it was first sort of shown in the 1940s, not very complicated. You expose people to low levels of oxygen, their pulmonary artery pressure goes up, and if you take that hypoxic stimulus away, it goes back down to normal. 20 years later or so, when people started doing studies on these residents of high altitude, they noticed that even giving them 100% oxygen did not bring their pulmonary artery pressure back down to normal. And then studying those individuals who were in the Operation Everest 2 experiment, they found that even with this relatively short exposure to hypoxia, even they did not return their pulmonary artery pressure back to normal given oxygen after that. And this is thought to be due to a degree of remodeling of the pulmonary vasculature due to this hypoxia. Mike has already mentioned Jim Millage previously, this sort of legendary high-altitude physiologist who we've had the pleasure of working with a number of times now. With Jim, when we went to the Margarita Hut, one very small study we did was to try and look further into this vascular remodeling of the pulmonary vasculature by simply just measuring pulmonary artery pressure using echocardiography and then giving people oxygen at the Margarita Hut, so about 4,500 meters, and measuring it again, seeing if we could get their pulmonary artery pressure back down to normal. But just to demonstrate how legendary Jim Millage is here, you may recognize, does anyone recognize who he's standing with? Sir Edmund Hillary and Griffith Pugh, so a true legend of the sort of high-altitude world. So these are the values that we got just for those 4 subjects, very small study. In the black dots, you can see the pulmonary artery pressure that we measured, just simply breathing air at the Margarita Hut, and then attempting to return our pulmonary artery pressure to normal, breathing 35% oxygen, and you can see that it never goes back down to the red line, which was our sort of baseline there. From that, you can calculate something called the normoxic efficiency quotient, which basically is a measure of how easily you can get someone's pulmonary artery pressure back to normal by breathing oxygen. And you can see with time along the x-axis here, various different studies now showing that the longer you spend at altitude, the more difficult it is to get someone's pressure back down again, demonstrating this vascular remodeling with our small study there as that red dot. In 2007, and published a decade later, we did a much more extensive study of this, taking the trekkers that went to Everest Base Camp and not only returning their oxygen levels back to normal by giving them oxygen as they trekked up to Everest Base Camp, but also giving them a hypoxic gas mixture as well. So they breathed the air that they're ambiently around them and a hypoxic gas mixture and oxygen, trying to look at the same sort of thing. And what we can see is certainly we're unable to return the pulmonary artery pressure back to normal, but also that there's no change in the responsiveness of that measure in the period of the 3 weeks or 2 weeks or so that they're ascending to base camp. So there are other things to think about with the cardiovascular system are the redistribution of blood, considerably more difficult to measure than simply measuring cardiac output and stroke volume, so systemic measures of cardiac function is what happens to the blood after it has left the left ventricle. We, in one of our very first experiments at high altitude back in 2003, we attempted to address this question using probably a dying art now, sadly, of gastric tonometry, where you insert a nasogastric tube, and in the end of it is a porous plastic so that the carbon dioxide in the stomach can diffuse across into the balloon, and then you can measure the amount of carbon dioxide in the stomach and compare that to a systemic measure of carbon dioxide, such as either arterial or end-tidal carbon dioxide. It's as you can see from this photograph, it's a reasonably unpleasant study. It causes lots of interest from the locals. There's a sort of younger and more rugged-looking Professor Grocott on the left there. He's sort of slightly shorter hair than he does have now. And so we conducted this experiment, and what you can see with this sort of normalized data is that the wider the gap gets between the CO2 in your stomach and your systemic carbon dioxide, the greater degree of malperfusion of the stomach, and this is at rest, and we didn't have very much in the way of sophisticated exercise equipment as we did, as you heard in the last talk in 2003. We simply had to give people a bag full of rocks to wear to slow them down a bit and get them to step up and down on another rock, measuring their heart rate as a measure of their work that they were doing. We've also looked at systemic oxygen extraction, because I'm sure, like me, when you went to medical school, they kind of taught you that when things go badly wrong in the body, it's okay, because you can just extract more oxygen from that which is going round. And if you read between the lines of the Operation Everest 2 data, that did not seem to be happening. So there's a very rich data set from Operation Everest 2 back in the 80s, and you can basically work out the systemic oxygen extraction when the subjects are exercising and there's no change whatsoever, even at the very highest altitudes. So what we wanted to do was to repeat that experiment that they did at the Margarita Hut, because it didn't make sense to us. We assumed that basically you should extract more oxygen as there's less in the environment as you're exercising. So whilst storm-bound in the Margarita Hut, we inserted central venous catheters and arterial lines into one another, and then conducted a series of exercise tests measuring oxygen from the lines that we put in, and sure enough, just like the Operation Everest 2 data, we showed that there was no change whatsoever in systemic oxygen extraction from rest up to maximum exercise on the cardiopulmonary exercise testing machines. And so finally, I'm going to return to tell you about the microcirculation, as I stated from the outset, that this may be the place where all the answers lie. As I'm sure you know, the microcirculation consists of the very smallest vessels in our circulation, are usually classified by those being under 100 micrometers in diameter, arterioles, venules, capillaries, where all of the gas exchange and nutrient exchanges occurs in our tissues. We have used a couple of different devices to measure the microcirculation, side-stream dark field imaging and instant dark field imaging, which basically beam a green light into the mucosa, and then you kind of can take films of the moving red blood cells traveling through the microcirculation. These are the kind of images that you get, and you can see each of those black dots moving through the circulation is an individual red blood cell, so you can then process these films and analyze them with software to look at the flow through the microcirculation and also the density of vessels in the microcirculation. This is a summary slide of what we found in 2007, studying the microcirculation of our cells, so lowlanders ascending to high altitude. And what you can see on the left is where it says GX. This is a measure called grid crossings, which is a rudimentary but effective way of measuring the density of vessels in the screen that you see. So there's a persistent rise in vessel density as you go to high altitude. And then MFI is microcirculatory flow index, and it's an index of the speed of flow of blood through the vessels in the films that you're looking at. And you can see that as you go higher and higher up towards Everest Base Camp, the speed of the flow goes down. And the red and the blue is those who went to very high altitude, the climbers, and the team who stayed at base camp. And you can see this very reduced microcirculatory flow index in the climbers after they have come back down and rested from the summit for a number of days. We also, as you heard in the previous talks, studied the Sherpas, and we were very interested to look at microcirculatory function in the Sherpas, as we can find no other real great differences between us and them in systemic cardiovascular measures. So we measured using similar equipment to 2007, this microcirculatory flow index and vessel density. And what you can see here, the Sherpas are shown in black and the lowlanders in grey. And what you can see in the red box is that, as we demonstrated before, the lowlanders have this reduction of flow within their microcirculation. But the Sherpas, not only do they not demonstrate this, but their flow actually increases. Further to that, this is showing the density of vessels in the microcirculation, and again we confirm what we saw before, which is the lowlander vessel density increases, but you can see in the black box here, which is with the red ring around it, this vastly increased vessel density in the Sherpas compared to the lowlanders. If you don't believe in statistics and you think it's all a bit of a fudge, on the left is a lowlander microcirculation at Everest Base Camp, and on the right is a Sherpa microcirculation at Everest Base Camp. No science required to determine the difference between these two sets of microcirculations. In summary, adaptation to hypoxia at high altitude. The wiring gets all messed up at high altitude with this huge sympathetic drive that doesn't go away. The pump works much harder and faster throughout the time that you're at altitude, and that also is sustained during the time that you're there. This leads to increased pressure and flow during that time. The pipes alter in diameter, some getting bigger, some getting smaller. And some of those pipes, the ones in your lungs, may stay altered for some considerable time. Flow gets redirected away from less important areas, primarily the gut, but perhaps more importantly than any of these things is that we see very clear differences in the way that the microcirculation works between different populations and different people at high altitude, and maybe this has a profound impact on the performance differences that we see between people at high altitude. Thank you very much.
Top Med Talk. Don't forget you can meet the Top Med Talk team, ebpom.org/meetings. Our next big event takes place between the 28th and the 30th of September. EBPOM USA Chicago Masters Course, a perioperative care practicum.